This protocol describes a novel and easy method for creating mesh-shaped engineered cardiac tissues derived from human-induced pluripotent stem cell-derived cardiac cells.
The flexibility of the tissue geometry and the scalability of the preserved tissue function are the main advantages of this technique.
The implanted mesh-shaped ECTs can restore cardiac structure and function in a rat myocardial infarction model, confirming its feasibility for use in cardiac regenerative therapy.
These techniques require reproducible human stem cell processing and tissue culture skills for consistent results.
Begin by cutting a cured PDMS sheet to the appropriate size to allow the sheet to be bonded with silicone adhesive to fabricate a 21 by 20.5 mm rectangular tray with 7 mm long, 0.5 mm wide, and 2.5 mm high rectangular posts at staggered positions. The horizontal spacing between two lines of posts should be 2.5 mm. Autoclave the tray at 120 degrees Celsius for 20 minutes before coating the mold with 1% poloxamer 407 in PBS for 1 hour. Then thoroughly rinse the mold with PBS and place the mold into one well of a 6-well plate. After cell lineage analysis, combine the cells from the cardiomyocyte plus endothelial and vascular mural cell protocols so that the final concentration of mural cells is 10 to 20% in a total cell number of 6 × 10⁶ cells per construct. Mix 133